好的，现在是晚上的8点，非常高兴我们今天晚上的直播就正式开始了。同筑技术人生路，写意中国新药魂。各位行业同仁，大家晚上好，我是同写意的张蕊，非常欢迎大家来到我们今天的写意直播间。那相信大家对我们的直播的一个背景已经有了一个了解啊。那我们知道，稳定性研究是贯穿药物研发到生产的多个环节。那传统稳定性研究耗时久，对长期稳定性预测性差，所以这是传统稳定性研究成为时间成本、机会成本最高的 CMC 工作之一。那为了解决上述的问题，基于风险的预测稳定性应运而生，特别是 ASAP 技术，以 ASAP 为代表的 RBPS 技术呢，已经在全球范围内数百个 IND 和 NDA 的申报案例当中得到了一些广泛的应用，并且在写入新版本 ICH-Q1 指导原则的过程当中。所以呢，同写意和南京海维医药是特别策划了为期3天的 ASAP 技术系列讲座。那今天晚上是我们本系列直播的第1场。那首场直播我们特别邀请到的是FreeSync 总裁、ASAP 技术先驱、RBPS 意见领袖 Ken Waterman 博士，还有我们同写意药学院院长肖百明博士，还有南京海维医药创始人王立坤博士。那3位博士将共同探讨稳定性预测原理应用监管和 ICH-Q1 更新趋势。那在直播过程的当中，如果您有任何互动问题，都可以在我们评论区留言，或者是点击我们直播间下方的提问来告诉我们。在分享讨论后的写意问答环节呢，我们会有请几位老师来进行解答。那相信大家已经非常期待我们今天晚上的分享。那我们看到大家纷纷在转发我们的直播啊，那我们大家可以 call 一波666，欢迎我们今天晚上所有的分享嘉宾。大家可以把666刷到我们屏幕上，欢迎我们的所有嘉宾。同时也欢迎大家都将我们的直播间分享出去，我们更多的人看到有很多观众朋友在支持我们啊。好的，那我们首先有请同写意药学院院长肖百明博士进入直播间。我们首先欢迎肖博。我们先邀请肖博。好的，那非常欢迎肖博。肖博您好，您先和我们观众朋友打个招呼吧。
您好，主持人，各位听众观众，晚上好。
也非常高兴啊，今天晚上我们这个首场直播是邀请到了肖博给我们坐镇。那肖博是这样啊，据我所知，其实我们业内了解 ASAP 的人并不是很多，但是 ASAP 对于药学研发，尤其是新药研发方面还是非常重要的。其实我也听您谈起过 ASAP 啊，似乎可以是用一首歌名来概括，是像雾像雨又像风。那么也想先了解一下，就是您是怎样理解的，然后对于我们研发人员又有怎样的意义呢？
这个实际上是一部电视连续剧的名字，差不多是快20年了啊。这个我不知道年轻的朋友们有没有看过这个电视连续剧。那时候我在美国，我觉得呢，这个 ASAP 模型呢，对我来讲呢，我的理解更像一首歌。那么歌的名字就叫让我告诉未来自己命运自己主宰。那么我想呢，今天我会用一个非常短的 introduction 来介绍一下这个方面的背景。那么是后面的呢，就是说我也是抛砖引玉，那么使得后面呢，可能对大家后面的几位 speaker 的这个演讲呢，有一个初步的理解。好，那么我就想，为什么我讲这个是让我告诉未来自己命运自己主宰呢？我们看这一页。那么这一页的话是一个3个色谱图的叠图，那么下面呢是1个月的时候室温的我们的这样色谱图啊，我们大家看比较这中间上面这个它的这个 degradation， 就是它的这个降解的情况，有关物质的情况。那么我们就想到这个药物在未来假设2年以后它应该是一个什么样的状况。那么你在1个月的时候你是很难想象的，对不对？它会产生哪些有关物质。那么假如我们能设计一个实验，那么像中间这个红颜色的框框 2个星期在60度，我们就能够产生这样的一些降解物。这个所谓降解物的途径 degradation pathway 就和我们未来2.4年这个上面的图谱几乎一致。如果我们在研发中间能够做到这些，那我觉得对研发人员来讲就是一个非常大的喜讯。然而我们并不是都是这么 lucky， 并不是这么走运。那么就我个人来讲，我有几段艰苦的经历。那么这个经历呢，在20多年前，我们这个大药刚好快要上市了。然而就是在上市之前，6个月的长期稳定性的结果出来了。那么就在每一个这个稳定性条件呢，都有不同程度的降解，尤其是在40度，75%的湿度，这个含量的降解呢达到5%，然而总杂却1%都不到。那么基于这样的情况，那我们是没有办法申报的。所以当时我是受命于危难，我解决了这个问题，一共花了大概快2个月才把这个 mass balance 的问题解决。那么我们怎么解决这个问题的呢？实际上当时就在想，因为我们到稳定性室的话，要这个同样的这个26个星期这个加速实验的样品的话，我们可能只能拿到几瓶，我们不可能拿到50瓶、100瓶，我们要做大量的 extraction test， 所以我们必须要在短时间内要产生就是一个 meaningful 的 sample， 就是非常有益的 sample。所以我们试了各种条件，后来我们就试了在85度高湿度的情况下，那么大家看到了上面蓝颜色的，它一天产生的降解物，那么大概的相当或者是超过于我们6个月稳定性加速实验的这样一个数据。那么这就是给我们自己产生了就是有益的 sample， 我们下面用这样的 sample 进行了一系列的实验，终于完成了我们对这个mass balance deficit 这样一个研究。那么第二个案例呢，就是我们当时在完成了这个实验以后，我们就发现了有一个不稳定性的降解物，后来慢慢发现它的分子量比这个 API 高了30，我们非常幸运，那么就认为它这个是一个甲醛的加合物。那么这个甲醛从哪儿来呢？我们从 API 和各种辅料反应，最后我们发现，那么这个是甲醛呢，是来自于欧巴代。那么在欧巴代的反应中间，我们在进行了进一步的研究。从这个模型，大家看到，从0时间到1个月，那么各种红的欧巴代、白的欧巴代，以及下面图里面含有各种辅料的这样一个这个反应，那么就是大概的差不多在0.3几左右。那么大家1个月以后都达到了饱和，就不再反应了，这也很 make sense。因为什么呢？这个时候甲醛残留，那么在辅料里面也反应完全了。那么就是我们建立这个模型呢，回过头来看看也有道理。那么30度的模型，因为我们在最初的话，我们也想用非常高的模型，那么后来最终发现30度跟我们实际的样品也是一致的，因为在高温条件下，甲醛就会蒸发了。那么另外还有一个案例，就是说也是一期临床以后，我们也发现了一个片剂的它的这个物料缺失。那么这个时候呢，我们就发现在样品提取方面是存在问题的。所以后来我们进行了很多的 study， 我们终于了解到，那么这个物料缺失呢，是从样品提取的不完全所造成的。那大家看下面2个图，分别是左边是它的含量我们用9个平行的样品，我们发现它的含量从老方法的 95.9 提高了到 99.2。 那么大家也看到，从这个标准差这个 standard deviation 也是有明显的下降，就意味着我们这个最后测得值朝这个 true value 非常接近的时候，它的精密度也大大的提高。不但这样，对于含量我们得到这样结果，那么我们对于总杂 大家注意，这个总杂质是什么得到的呢？就是大量的是位置杂质，因为我们对这个样品进行了湿热的破坏。大家看到又是一个模型，75度高湿度24小时的模型。那么它产生的降解物呢，是远远高于我们临床我们这个稳定性实验1年的数据，所以我们就这个实验也昭示着，即使我们这个项目进入到3期，我们有做到心中有数了。那么这个原理是什么呢？这个实际上就是我们对于这个研究温度在高温下的破坏是按照阿伦尼乌斯公式，也就是公式的左边。那么我们刚才看到了湿度也产生了比较重要的影响，所以下面第2项就是这个 B 温度敏感系数乘以平衡相对湿度这样一个公式。那么这个公式呢，是我们下面几位 speaker 在演讲的时候都要经常用到的这个公式。那我们现在看这张图，上面一个降解物 B 在这个 amorphous 就是一个无定型的这样一个材料里面，那么大家看它的纵坐标是降解物的含量从0到 0.1%， 它的横坐这个平面坐标呢分别是温度和这个水含量，用水含量呢不同水含量呢来控制它的湿度。那么不单单我们在有这个含量和有关物质上面，我们用了 ASAM 模型，我们可以做很多有益的 study。那么在没有产生严重的降解物的，我们也能够监控我们的物理变化。这一张图呢是左边，就是我们在各种湿热条件下不同的时间来看我们各种湿热破坏的样品的溶出行为，溶出这个曲线和蓝颜色的这个没有破坏过的这样的一个样品的对照。那么大家看右边这个图，就是分别在不同的温度、湿度实验设计点和这个不同的时间。那我们最终的目的是什么呢？我们的目的是 target 这小的绿框框里面的，那我们就是要什么呢？在25度到30度，我们一般的储存条件下，在60%到70%这个湿度，那么比我们室温条件再破坏性更强一点。我们展望2年后、3年后，那么这个货架是否能够还满足要求？那么下面这几张 slides 呢，我是跟王博借用的，我想用来之后呢，作为一个铺垫，这也是抛砖引玉。这样下面呢，给我们的大家观众、听众呢，做一个初步的了解，以便于我们下面更好的理解我们各位 speaker 讲课这个演讲的主要内容。那么左边我们看这个图呢，是红颜色的标刻75度的，那中间是60度的，下面是黄色是50度的。那么它怎么会变到像右边这张图呢？我们可以想象，我们用一把像钢丝一样的把它们抓起来，凝结在一起，大家看到了。那么这些破坏的条件，那么它产生的这个 degradation 的这个量以及所需要的时间都是不一样的。好的，我刚刚所提到了，那么这个原理呢是基于我们阿伦尼乌斯公式前面一项，那么还有后面一项就是 B 乘以 RH。那么这个里面有2个非常关键的参数，对于温度来讲，我们要重点理解的是它的反应活化能。那么这个反应活化能越低，这个反应越容易进行。那么如果活化能越高的话呢，越困难，那我们就放心了，对吧？那么下面一个呢，这个后面的湿度敏感因子也是的，如果它低，就表明它不敏感；如果是高的话，我们就要 pay special attention。那么下面这一页呢，就是讲活化能。我们通常是在17个千 K 摩尔这个 level， 我们用这个半年的40度的加速实验来预期我们2年的室温模型，根据阿伦尼乌斯公式。如果这个活化能是明显高于17甚至20几、40的话，那么这个高活化能呢，就是说，那么这个化合物也可以说它是稳如磐石，它也不可能反应。那么下面呢就是一个温度因素，那么温度因素它分为3个水平，下面的0代表是低这个敏感性， 中间0.04是代表是中间的这个敏感度，0.09是 high sensitivity。那么这个里面一个表的换算也是在不同湿度下需要不同的时间的换算，我想我这儿就不多讲了。好，let Dr. Waterman, I apologize. I spoke Chinese because most of the audience native language is Chinese. Now it's my great pleasure to introduce Dr. Waterman. Dr. Waterman got his bachelor degree in University of California in Los Angeles, and also he got physical chemistry PhD in UC Berkeley. He had many experience in Florida and Pfizer. After 2011, uh, he worked, worked in this area. So as a president in Free Think Technologies, he is a great pioneer and a great leader in this aspect. So let's welcome Dr. Walkerman. All yours.
Let me see if I can share, hopefully. Um, won't let me share. Ah, no, it won't let. Yes, it's this one. Ah, I'm so sorry. It it won't let me share. It did before, but now, sorry. Okay, I think I've got it. Um, Okay. Sorry for everybody. Thank you for the introduction, and let's see if I can find an angle that's good. Okay. So I want to talk about stability modeling. So we're not really predicting stability, we're actually using data to predict stability. And so we're going to be doing modeling as part of this. So I'm going to go through a little bit of what we call ASAP or ASAP to determine drug product or drug substance shelf life and how that works. So we'll go through a little bit about some of these principles. I think you heard a little bit of that in the introduction. We'll be talking about ISO conversion, why we use it, what it means. We'll go into a little bit about moisture sensitivity, probably not a lot on statistics, packaging, a little bit about regulatory. So ASAP Prime, which is the software that we produce that's now used by almost all the major global pharmaceutical companies, involves exposing samples to humidity open. So this is for solids. For liquids, we don't need to do that. So what you see is we have a sample. It could be tablets, capsules, powders, or almost any kind of product. We put a saturated salt in a vial with a moisture permeable top, and this is all put into a what we call in English a mason jar. So it's a jar that we use for making jams and things like that, but this works very well. So the sample is open to the humidity, and we use this because it provides a good, let's say, mini chamber for the sample. We measure whatever is shelf life limiting. So this could be chemical stability, as was discussed, could be formation of degradation products or loss of assay, but it also could be color or dissolution or physical form, pretty much anything that determines shelf life has, we say, activated process. And then we're going to take these experimental data and model it using ASAP. So what we do is we do the determination of the shelf life in this open condition, and then we predict what will happen in any kind of packaging by determining the humidity as a function of time inside the packaging. So the advantages of this approach, it gives us a fundamental understanding of temperature and humidity dependence. In fact, we also can do oxygen, but that is more difficult. This allows us, without having to do any package screening, to determine what will happen with a change of packaging or different packaging, and also determine what will happen if there's an excursion. We can also help solve many stability problems by a better understanding of what's going on. We get speed. So a typical ICH approach could take more than 6 months to determine shelf life. Typically, an ASAP study might be done in 4, 5, 6 weeks, so significantly less time. So what is this isoconversion approach and why do we use it? So what happens is over 50% of drug products show, we say, complex kinetics, which means that the assumption of linearity is not a good assumption. And here are just some of the reasons, and I'm going to show you a little more detail about these reasons. So let's start with the first one. This is one I call heterogeneous kinetics. And what that means is, if you kind of imagine the world of a drug in a drug product, you have molecules of the drug that see different microenvironments. Some molecules are locked in the crystal lattice, and when they're locked in a lattice very tight, they don't move much and they're very stable. Some molecules are sitting at the surface of the crystals, and they're going to be a little bit more mobile. And then there's even some that are in either solid solution where they interact with the excipient to form a mixture that is now no longer crystalline, or pure drug, but amorphous. So both of these two are going to be orders of magnitude more reactive than molecules in the crystal lattice. Each of these has a distinctive rate constant, a K. They may each be very simple, but when we go to analyze the samples, all we get is the superposition of these individual rates, and the result is we have a kinetics that is very complex and not described by any simple curve shape. Another example of this, well, this is what that might look like. So the inside the crystal might be the bulk of material, but very slow growing. And the material right at the interface may grow very fast, but maybe there's not that much of it. And the overall kinetics becomes this complex curve, which cannot be assumed to be linear. Another example of this that's pretty common, even in liquids products, is secondary degradation. This is where a drug goes to a primary degradant, and then the primary degradant goes to a secondary degradant. So the primary degradant, which is this lighter blue color, the primary degradant grows more quickly, but then as it starts to build up, it depletes and it curves off. The secondary degradant lags because there's nothing in the beginning of the primary degradant. So both of these are not well described by a simple kinetic model. Now, I mean, they can be described by fancier kinetic models, and you have to do that sometimes. But in either case, most simple approaches will not solve for these. So traditional accelerated aging is not predicted of real time, especially when you use high temperatures because of this. And since we can't use a simple linear approach, what are we going to do? So can we use kinetic modeling in the absence of a detailed kinetic study? So we're going to see the solution is this isoconversion. So when we do traditional accelerated stability and we try it on something that's very nonlinear, if we imagine this situation where we have a very nonlinear kinetic growth of a degradant, and we looked at it at 60 degrees and 70 degrees for, in this case, 3 weeks, we can do what people would traditionally have done with accelerated stability and assign a rate constant, or K, based on the amount of degradant formed divided by the time. And we're going to get kinetic rates, and certainly the rate is higher at 70 than at 60, and certainly very different than at 30. But you'll notice that the rate at 70 and 60 does not match the rate at 30 very well at all. So this results in the appearance of non-Arrhenius behavior, non-predictive. And this was why prior to our work in this field, people often thought that when you went up in temperature, you couldn't predict stability accurately. In fact, even in the ICH, it says things like this. But it turns out we can change the approach and now make it very predictive. So what we do here in isoconversion is, instead of focusing on keeping time as the constant, we keep the amount of degradation constant, or isoconversion, amount of conversion, the same. And where are we going to focus? We're going to focus on the specification limit. So in this case, I've assigned a specification limit of 0.5%, which is a typical amount of a degradant at the end of shelf life. And I find the time to fail, or isoconversion time, and I now have a different K, a K representative of the time to fail. So the amount of degradant stays the same on the top, and on the denominator, we change the time, and now we have two different rate constants. Now when we do this, the predictions become very Arrhenius. So by using this isoconversion approach, with isoconversion, we can make accurate predictions from high temperature, even if we don't know or cannot solve an exact kinetic form. So what we do in an ASAP study is we try to shorten the time at the higher temperatures and use longer times at the lower temperatures, and try to determine where the product fails at each temperature. So we actually want it to fail at every temperature. And as we'll see, we add this humidity term. So the humidity term, which was already presented, allows us to correct for the humidity. So I'm going to skip what was already shown. This equation mathematically is the equation of a plane. And so what we're doing is we're predicting behavior out at, let's say, 25 degrees, 60% humidity. And we're going to use temperatures and humidities in this upper temperature region to project where we are down here. And if we have enough data, we can solve this plane and determine the slope here and the tilt angle here, which will represent the temperature sensitivity, or activation energy, and humidity sensitivity, or B term. Now, the part, I'm not going to go into detail, but the software puts in also a lot of statistics. So the statistics are such, you can sort of see the higher the temperature data we work with, the greater the extrapolation to the storage condition, whether it's refrigerated or room temperature. The greater the extrapolation distance, the greater the divergence of the error bars. So what this means is we can make accurate predictions on short times, but we're going to pay the price in greater error bars. So the longer we're willing to wait, the longer we wait, the lower the temperatures we can work at, the less extrapolation, and the tighter the error bars. So we're always going to be trading off. If we're building a stability model, if we can generate more data, better data, we can make up for the fact that we are increasing error bars. We try to get our analytical methods more exact than they need to be in traditional stability. So one of the things we do in an ASAP study that's very different is we actually analyze all the samples in one batch. That reduces the error because typically the analytical methods have greater error run to run than within run. So we reduce error. We try to be very careful on designing the studies so that we can get the precision we need to determine the shelf life. So we talked about using these mini chambers. Another reason we like these is we can use one oven and get multiple humidities. We can have samples. If we tried to put samples open in a humidity controlled oven, we would have the risk of cross contamination. And also we typically equilibrate the sample to the humidity overnight before we adjust it to temperature. So we can make the humidity become at equilibrium before we go up to temperature. All right, now that was determining the humidity open, temperature sensitive, humidity sensitivity are known. How do we determine the humidity that a sample will actually see? So whether we're in a bottle or blister or any other kind of packaging, there's going to be humidity outside, water inside. If we imagine inside a package, we have dosage forms and we have maybe a desiccant. Inside the packaging, everything will come to equilibrium fast compared to the time scale of moisture coming in or going out of the package. So moisture typically will then enter the package based on the moisture vapor transmission rate, or MVTR, and the difference between the relative humidity inside and outside the package. So the moisture will equilibrate inside based on the moisture sorption isotherm of the product. So relative humidity as a function of time is very exactly predicted based on the MVTR, the moisture sorption isotherm, the headspace volume, which turns out to be a relatively minor component, and then the external conditions. So all of these allow us to predict humidity as a function of time very exactly. This is just some example of predicted versus measured. In general, this is not new. This is something that, the engineering equations for this have been around for quite some time. Very exact. So we have our stability model. We've determined our temperature and relative humidity dependence. We determined the relative humidity inside the packaging as a function of time at the temperature. Now we can determine the stability for a given package. So just to look at a couple of examples, here's one that was published. It's a liquid, so we don't have humidity dependence, but the prediction interval is between this green and red. The blue line is the mean prediction, and the squares are what were actually found over 3.5 years for this product at 25. So using a 3-week ASAP study, we very accurately predict what happens over 3.5 years. Here's another example. This was a tablet product stored 3065 for 3 years. Again, the 3-week ASAP study predicts very exactly. Again, another one. This is a tablet in a bottle with desiccant. The red line is the prediction. It's quite a stable product, but the original specification limit was only 0.2%. So there wasn't much room for variation, but the predictions were very exact for 3 years. This was a product we did that's a nicotine-based product. So this one was not degradation formation. This rather was a loss of nicotine. The nicotine is in the form of an ion exchange resin complex, dissociates with time, and the packaging has a huge, very large impact. This particular packaging was polyvinyl vinylidene chloride blister package. This was the shelf life predicted by ASAP in these 4 storage conditions and what was actually observed for the shelf life. So very good matchup. What it doesn't say in this particular poster is what would be needed to get the shelf life in other zones. So in this particular case, the cost of packaging is more expensive than the product. So there's always a balance between getting better shelf life versus paying more money for packaging. This one was a peptide, loss of peptide assay. So this is the peptide bacitracin. And again, you could see very good match between the prediction interval and what was found at 3 different storage conditions. This one was done by people at Johnson & Johnson in Belgium. The blue line is the mean prediction. And again, the prediction interval is the error bar from ASAP. 2 years, again, a gelatin capsule product, so very well predicted. And this one, AstraZeneca, this was a refrigerated IV solution, intravenous injection solution. 5 degrees was long-term, 25 degrees was accelerated, and both were predicted very nicely within the confidence intervals. Just some examples that were done by Pfizer, just pointing out many different mechanisms, and yet all predicted very nicely by the ASAP models. So right now, there have been hundreds and hundreds of examples, predictions working very nicely. I'm going to also mention that this has now been used fairly extensively in regulatory filings, but this was a few years ago. There was a survey done of large pharma companies that used ASAP for clinical trials in different regions of the world. There have been many since then. What these were only using ASAP to assign a initial use period or expiry, depending on the country. And in each pair, sorry, I touched something. In each pair, the red is the number submitted. The green is the number of those that were accepted with no requests for further data. So as you can imagine, the US FDA is very accepting of ASAP. Belgium has generally been. In China at the time, there had only been one, and it was accepted. Now there have been more in China, but at that time there was only a few. So I'm going to show you a little demonstration of the software, but before I do that, I just kind of to conclude, accelerated stability can be applied effectively to set shelf life of drug substance, drug product, using ASAP Prime. I mean, we're just to mention some things you wouldn't even realize. For example, we're going to be publishing now a case study showing that we could use ASAP to predict the shelf life of probiotics, which really amazed me that worked. But probiotic shelf life can also be worked with ASAP. So we use 3 principles in developing our ASAP. We use this isoconversion where we focus not on the rate, but on the time to hit the failure point at a range of temperatures and humidities. We explicitly then count for moisture sensitivity. So not the external moisture, but what the actual product sees. We build in error bars, so we don't say what is the shelf life, we give what is the probability of passing. So most companies accept as long as there's a 95% confidence, that's acceptable, and the health authorities have generally accepted that. And with that, we're able to make better decisions faster. So let me just show you. Are you seeing the software? Yes? Yes? Okay. Assuming you're seeing this, let me just show you kind of example. So this product is, you're not seeing. I was afraid of that. Okay. So I think it only allowed me to share the presentation. So maybe what I'll do is allow for questions now then, because it looks like I wasn't able to share the screen.
是的，我们现在看到Dr. Waterman 您这边的屏幕共享是已经结束了啊。那也非常感谢我们 Dr. Waterman 和我们刚刚肖博的一些专业讲解。那现在呢，我们邀请同协医药学院院长肖百明博士和南京海卫医药创始人王立坤博士，我们一起进入直播间，共同和我们观众朋友进行一些互动交流啊，我们答疑解惑一下。好的，看到所有老师都上来了，肖博好，王博好 ，Dr. Waterman 晚上好。好。那其实我们现在评论区里面已经有观众的很多问题啊，那我们接下来也对这些问题呢再继续的探讨一下。观众问到第一个说，请问 ASAP 在生物药或疫苗当中的应用前景如何？那是否被监管机构认可呢？
这个问题我先翻译一下，然后呢，同时我也可以做一些基本的解答。So the first question is, can ASAP be used for biologics or vaccine? And what's the future of regulatory towards ASAP kind of tools?
Okay, let's take one question at a time. Okay, so the first question is, can ASAP be used for biologics? So the answer is somewhat complex. So let me kind of take this in stages. ASAP works fundamentally for anything that stays in the same, same phase space. That means that if you have a biologic and it's going to be frozen, below freezing, you can't really predict the stability from above freezing to below freezing if it's got water present, because the freezing of the water leads to a discontinuity in the behavior mathematically. Those are hard. Second, if you have a liquid and stays liquid, let's say it's in water and it stays in water, there can be phase transitions between the different phases of the biologics. Those can make a discontinuity in the stability behavior. Generally speaking, ASAP will work well on biologics for specific biological mechanisms that are, or chemical mechanisms of biological molecules. So if you have a specific, let's say, deamidation, should be fine. If you have a, let's say, a drug antibody complex, the dissociation of the complex will be fine. When you have biologics that are small enough, like peptides or smaller proteins, where most of the stability is associated with primary degradation, should be fine. When you get up to things that are big, then you start to have issues of what is really going on stability-wise, and those can make this more difficult. So there have been a number of examples of successful use of ASAP for biologics, but it's not easy. The other thing that makes biologics very difficult is analytical methods are very, very high error bars, big error bars. And we're going to make error bars bigger by propagation. So if you don't have a good analytical method, so for example, bioassays are very noisy. And if you try to accelerate, you're going from noisy to even greater noise. It's almost meaningless. It's almost meaningless even at the current condition, never mind accelerated. So these are the considerations with biologics. So the answer is yes, maybe, sometimes, but you need to do it. In English, we say with your eyes open. Okay. I don't know if you have a similar expression in Chinese, but eyes open for it. Okay. Your second question was what? I lost it.
It's about
regulatory of some kind.
Regulatory's opinion towards tools like ASAP.
Well, again, this is difficult, a good question, but let me try to answer. First of all, there is a global effort in the International Convention on Harmonization, or ICH, to revise the guidelines to allow for ASAP. They are expected, that now has an expert group working on the language, and the expectation is they will come out with a revision to ICH around 2 more years. That's approximate. So the good news is that the international community has agreed that this is something they want to revise, and this is one element of the stability revisions. There's other elements as well. Now that, first of all, means ICH countries. There are many countries that never accepted ICH. Second, the health authorities it's not a simple question for them, it's which reviewer looks at an application. We have had a lot of success in many countries. Certainly, the countries that like science tend to be good. The countries that like rules tend not to be as good. And there's some that are in between. Like we would say Germany and Japan like science, but like rules. So sometimes they're more complicated. So the answer is will depend a lot on what you're trying to do. We have found that adding the modeling with some traditional data can be very effective. Making the story a good logical story. It also depends. Is this a drug that is helping a critical medical need? Is it treating illness, a pandemic, or something there's not a lot of other diseases cures? That's going to be better. The other side, is it a drug for which there's many years of data and you're making a change, but there's easy to validate the ASAP model because you have lots of years of data. So each case is something you have to work on, but there's no statement from the health authorities. They simply either accept it or ask for more data or more explanation. There is not a unified, the FDA says. The FDA is hundreds of people, hundreds of reviewers. Each one may say something different, but we have had a lot of success in all phases with them. But depends on the country and what you're asking. Were there other questions?
我们问题还是比较多的啊。那关于这个问题，我们王博和肖博，有没有什么补充呢？
我来补充一点，就刚才科莫特曼博士提到了，就是比较重要的是这个 ICH 现在正在是这个 Q1 正在做一个 revision 的一个过程中，就正在升级。那么以 ASAP 为代表的一类稳定预测的工具的话呢，大概会在2年左右的时间里面会正式进到新的版本里面。那么在这以前的话呢，科莫特曼博士提到了，就是说在这个很多的国家里面，特别是欧洲、美国的这个药监部门里面，已经有比较多的这个成功的一些申报案例啊，那么特别是这些对于这个急需上市的一些品种啊，那么包括这个之前辉瑞这个新冠的小分子药 Paxlovid 也是用的 ASAP 技术的话呢，这个很快的拿到了1年的货架期。那么在
对这个屏幕共享一打开，我们的画面。
Now should be OK。
对，现在 OK 了。
Sorry,
好，那我们还是回答这个问题啊，我们刚刚观众没有听到的部分。
It's all in Chinese.
第二个问题的话呢，就是我们提到了这个对于生物药到底能不能用，是吧？那么因为我们大伙儿都看到了，因为 ASAP 呢，它是基于这个化学降解动力学的一个预测的一个工具啊。那么所以这很取决于呢，这个生物药呢，它的这个我们关注的这个货架期呢，它是不是基于化学的一个原理啊？那么刚才沃特曼博士提到了，像这个具体的这样的一些个从二聚体啊变成单体，或者单体变成二聚体过程，那么这呢是有可能是可以的。但是如果说是在生物药的存储过程中涉及到一些相转变的一些过程，那么比如说呢，从这个冷冻的环节到这个常温的环节，或者说呢，很多蛋白药呢，它本身是有一些个比较高的温度，它会出现一些变性的一些另外一个项的一个特征。那么这时候的话呢，它这个是不适用的。那么在有一些分子量比较小的一些多肽药物，那比如说多肽，那么其实它呢很像一些个这个小分子的属性。那么这个时候的话呢，这个 ASAP 是可以适用的。包括 King 刚才也举到了一个多肽的一个稳定性的例子啊。所以呢，这个问题的话呢，应该是 case by case 的这样的一个结论吧，需要大伙儿的用一个这个比较科学的一个眼光去这个判断啊，哪些时候可以用，哪些时候不能用啊。
好，那肖博有没有什么见解要跟我们分享的？您可以打开您的麦克风。
肖博，您那个在静音，
开一下麦。对。
刚才这个 Dr. Waterman 在解释这个上面解释的非常好，他也提到了许多国家在考虑这个 rule 跟这个 science 两个方面的 balance。那么前面的这一段呢，这个王博翻的也非常好。那确实是这样，有一些大分子的，它都要在低温冰箱保存的，那么你怎么样来做这些实验呢？那是不可能。而且我们蛋白的这个刺激结构，对吧？你破坏了它结构，它整个变形了。就作为我们搞小分子一个 common sense， 对吧？你可以想到你怎么样跟小分子比较接近，对吧？刚才这个 Waterman 提到这个多肽，它比较类似于小分子的研究，就比较容易来做我们这样的实验，对吧？所以我想有一些是冷冻保存的，你这个加速的做不起来，这个状况下就很难做。那前面也讲到 ICH 指导原则，对吧？在近来的就是这个版本啊，未来的这个 version 上面来考虑，就是说 include 这些东西。但是我们要谨慎的乐观看到，那么这些东西呢是一个 scientific tool， 就是说也是我们怎么样来预见这样未来，这是非常非常好的。那么我也刚才讲到了，我举的几个例子也是没有办法的办法，急中生智，找了一个就是 very close to 这样一个这个 environment。但是对于我们今天这个 SAP 模型以及来讲的话，这个模型已经是我认为是非常好了。这个年初的时候啊，我跟王博也聊到了，他们特别在有一些这个坚如磐石的这样一个产品当中，就是我们不能够 generate 这个就是这个 enough 这个 dividend 的情况下，我觉得他用那个最小的模型，我们下面不知道有没有这样的东西，我觉得是非常好，就是最差的一个 scenario， 我们在这样情况下能够解释的，我觉得他是用一个就是一个这个统计模型，对吧？数学模型来做这样一个东西，是非常好。那么就是这一个 science 和这个 rules 这样一个 balance， 我想我们呢不要太寄予希望。那我们做了这个东西，好像我们这个长期稳定性都不要了。大家想一想，就我们连做这个加速实验6个月下来，它也不代表说我们就不要做长期稳定性实验。我们3个批次的报批这个承诺，对不对啊？那么它虽然就是说根据 ICH Q11 这个 Stability Data Evaluation， 我们最长如果有6个月的这个稳定这个加速实验，对吧？而且它心脏没有变化的情况下，我们常温在那个时候只能追到 2x， 对吧？也就是1年。那么这还要是 continue 进行你的这样一个 stability study。所以我觉得主要就是尤其我觉得对我们一个新药的研究，这是一个非常非常好能够展望你未来这个品种 it looks like， 大概就是这样一个，我是做这一点补充。
对的，同意肖博讲的，特别是肖博提到了在新药研发的过程中呢，有很多的不确定性。那么 ASAP 的话呢，它更多的是这个节约大伙儿开发新药稳定性研究的这样的一个时间成本的一个问题，是吧？就是我们不用等那么久，但是呢，大伙儿这个不要在短期时间内寄希望于它能够取代长期稳定性。我觉得长期稳定性还是一个金标准，就是说不会被取代。但是我们有更多的、更快的工具，能够看得到、展望得到那个以后呢2到3年的时间里面会发生什么事情，就不用等那么长的时间。
好，那其实我们几位老师给剖析的是非常深刻啊。那我们时间关系，最后再来提一个问题吧。我看到大家一直在问说，Can ASAP replace traditional stability study in our CMC dossier？ 这个我们先请 Dr. Waterman 来回答一下吧。
So we got another question. Can ASAP replace traditional stability study in our CMC dossier?
Well, the answer is typically what we see for companies is rather than replace, it becomes, we say, supplement. What I mean is that a lot of companies might only have 3 months of traditional data and still ask for 2 years of shelf life. They'll use that in conjunction with ASAP, and then they supplement the dossier with additional traditional data as it comes forward. So this allows basically they commit to updating. If something deviates from the trend, they would have to change their application. So it allows them to get product produced and on the market earlier, but does not increase the risk that unstable product will be on the market. So this balance between helping to get a product out to patients, helping medical needs, but also managing the risk is part of the approach we use. So we don't see a lot of applications where it's just ASAP for the regulatory filing for a market application. For clinical, yes. But for market applications, they're usually a combination, but the combination can be very effective. Okay, so you predict, you say, here's what we're expecting, here's what we see. We have 3 months, it's predicting correctly at 40 degrees and 4075 and at 2560 and maybe intermediate. And here's now we're going to get a 6-month point. We'll update it. If it's still trending correctly, we're going to get a 1-year point by the time it's on the market. If it's not correct, we will come back to the health authorities. Okay. Obviously, different health authorities, depending on the country, will view this differently. And it depends. If this is helping a lot of people, the argument is easier. So the orphan drugs or critical medical needs, it's easier to make these kind of arguments. So for example, in the United States, Paxlovid, which is a treatment for COVID, was approved with very little data plus ASAP, but got supplemented as the data became available. So the recognition was the risk of ASAP being not perfect compared to the risk of people dying from a disease. The balance was easy to make. If you're making a treatment for something where there's many drugs on the market already, maybe the argument is a harder one. Okay?
好，我看到悟空说他的问题好想被回答一下，那这样我们辛苦几位老师再加一个问题吧。我看悟空你提的问题是，请问对于在温度升高产生的与室温条件下不同的降解路径的情况，那 ASAP 是否仍然适用呢？对，这个是悟空提出来的。
Let me translate. So the question is about when temperature increases, the degradation route got changed compared to room temperature.
Yeah, never.
Is suitable?
Yeah, so that is something that many people think. We've looked for that, hundreds and hundreds of products. What we see, why people think that, I was trying to say, why is this believed? If you look at the product distribution, the degradant distribution, it certainly changes with temperature. But that's each individual degradant has, let's say, a different activation energy, different temperature dependent. So at high temperature, some will become more dominant. But if you follow each individual degradant, we really don't see a lot of times that a mechanism will completely change as a function of temperature. In other words, if it's a hydrolysis, it's still a hydrolysis. An oxidation, still an oxidation. It's not as common as people might think. So if it did change, what you would observe would be nonlinearity in your Arrhenius behavior. And the software would look and say, you can see that there's some kind of change that occurs, and we would now know that, we cannot use the highest temperature. The other thing we do as part of the statistics on the software is it's weighted such that the lower temperature behavior is more heavily impacting the stability prediction than the highest temperature. That being said, we've not seen any clear evidence of a change of mechanism from drug to degradant. The distribution definitely changes, okay, but not a change in mechanism. I think that's something that is very difficult to find. It's possible in a university world where you say, I'm going to find something, you can find an example. In hundreds and hundreds and hundreds of drug products, we've not seen any evidence of it. So maybe, but I don't think it's common.
好的，感谢我们老师的解答啊。那最后看肖博和王博，咱们这边针对观众的问题还有没有什么补充？对，一是可以看看我们观众的这里面有没有想特别回答的问题。另外呢，就是针对
我来稍微补充一下啊，就是这个刚才这个 Dr. Waterman 讲的，他就是说我提到2点，就是说一个就是根据这个阿伦尼乌斯公式，某一个反应它有它的活化能，那么后面就实际上它里边这个对湿度也有敏感系数，那么它对这些数据也是在模拟，如果它明显发现这个里边的这个推断，就是预测这个模型上面的预测出现非线性的时候，它也能够告诉这个模型本身告诉，那么这个里面的差别。所以我想刚才这个 Waterman 提的大概是也提到这个方面的内容。还有一点呢，我想大家就是说不要有一个想我这个实验 ASAP 就能取代我的一个这个正常的我这个稳定实验。那么我今天我想不管这个 Q1 怎么样的来 Q1 我们最终版本怎么样来讨论这个问题，那么我不觉得这个东西就可以代替稳定，但是我们可以在 in the very beginning， 我们可能刚才我讲的，让我告诉未来我自己命运自己主宰。我们在发现这些反应的时候，对吧？我们预计到有这样的反应，那比如说我们裸饭的溶液或者制剂有这样的反应，那我们在研究按照 ICH 这个里面，我们可以就是说它的内包装、外包装，对吧？我想至少在温度上面不是有这么有效的话，在湿度上面我们一定是能够有效的，就是说来预测一些未来的数据。我想对我们将来真正的到了2年以后的这个长期稳定性的实验数据，我想我们有很多工作就在我们的研发期限做掉了。刚才 Waterman 也提到，那么这个我们这些工作是在 clinical study 阶段就要完成的，而不是到这个最后的阶段。我想这点我再 emphasize 一下。
对，刚才的问题我再补充一点，就是关于 Waterman 博士他提到了，就是我们在高温下见到一种降解的路径和这个常温下面见到的降解路径不太一样。他讲呢，其实这种现象在工业界里面非常非常少见，其实我在原来的工作经验里面也发现非常少见。但是有一种情况的话呢，可能是会被误认为是降解机理的不一样，就是多级降解的情况。这个话题的话呢，就是说我们在 周二的时候，上海强生的苏博士他会专门有一块介绍这一块。但是呢，比如说我们有这个1级降解产物，它本身不稳定，会再次降解成2级降解的时候，这取决于我们观测的是1级降解产物、2级降解产物，是吧？那么如果说是这个1级降解产物呢，它可以由2级的这种降解动力学决定它的浓度随时间的变化，那么它可以呈现出来和温度啊负相关，或者和湿度负相关的一个总体的特征。那么这样的话呢，可能和我们常识可能有点相悖，但是从机理上的话呢，其实是可能的。我觉得这种的可能性会更大一些。
好的，那我看到其实我们观众朋友说的也是谢谢我们各位老师的一个专业分享，特别实用也很中肯。其实我们大家在刚刚的回答问题当中也是得到了更多的一个信息量，同时呢，我感觉也是提振了我们的信心。再次也感谢南京海卫医药对本系列直播的大力支持。那时间过得很快，今天晚上我们的直播就要接近尾声了，我们也再次感谢各位老师。那我们有请几位老师点击一下结束连麦，我们后台可以休息一下了，谢谢。辛苦所有的老师。那最后一点时间呢，也预告一下我们下期的直播。下期就是在明天，也就是我们下周一，时间段改为下午的3点到4点。这个是我们 ASAP 技术线上系列讲座的第2期。我们邀请到的是百济神州小分子分析研发负责人高阳博士，和我们来分享预测性、稳定性及其在药物研发生命周期中的应用。那欢迎大家预约我们明天下午3点时间段的这个第2场的直播。另外，如果您没有及时看到我们之前的直播，想反复学习老师在我们直播过程当中的一些干货，那也可以点击直播间右侧的添加助手，我们可以邀您进群来继续的交流探讨。另外，大家可以观看我们的直播回放。那直播回放是在微信小程序上，而且是只有在我们微信小程序上才会有，所以大家可以搜索同写意即可获取我们全部的直播内容。那最后大家也可以关注一下我们同写意的官方微信号，第一个是同写意微服务号，后续我们写意直播的宣发主要会在服务号上来进行发布，所以有想多关注直播的朋友，大家可以关注一下同写意微服务这个号的动态。那第2个是我们同写意的订阅号，主要是做行业媒体，发布我们行业的一些重大新闻，还有行业分析的重磅文章。那第3个是写意宣发号，主要是发布中国新药行业内的大事要事，重点关注企业的融资情况、人事任命，还有国家发布的一些行业新规、制度、政策等方面的内容。那第4个是同写意 Biotech 号，也欢迎大家来多多关注。最后，我们行业的朋友如果有什么企业宣发、媒体合作需求，也可以加一下页面右下角我同事的微信来进行咨询。好了，那最后呢，我们也再次感谢所有的老师，同时也感谢我们观众朋友的一个陪伴。那我们就下期分享，明天下午4点见，明天下午3点我们直播间见，谢谢大家。
写意是中国绘画的表现手法，随性潇洒，行简而易分。2004年10月，一个寓意同写意的论坛在北京成立，并于2012年升华为同写意新药英才俱乐部。同筑技术人生路，写意中国新药魂。这是同写意的初心，也是同写意成立10周年活动的主题。10年的默默耕耘和专业坚守，使得同写意在医药行业开始崭露头角。从那以后，同写意陆续在北京、华东和华南设立专职机构。同时，制药化学反应和工艺专家委员会、BD 俱乐部、临床研发俱乐部也相继成立，围绕医药研发创新的各种主题论坛接连成功举办。从樱花烂漫的武大校园，到禅味十足的拈花湾，从天堂苏州的独树湖畔， 到道教发源地青城山，从宝岛台湾再到大洋彼岸的美国，童写意的足迹遍布祖国的大江南北，并且迈向了国际。仿制药一致性评价，中药经典民方开发，医药项目的合作与投资并购，新药中美双报，4+7药品集采，每一次医药政策巨变，每一个行业热点涌现，童写意都会勇立潮头，在第一时间汇聚一线一流人才，为产业领航，帮企业赋能。高山流水遇知音，彩云追月得知己。上百期的论坛活动，一次次童写意人相聚，平时的交流，各抒己见，尖锐的碰撞，启迪灵感，描绘出一幅幅形神兼备的新药研发写意画。在医药的圈子里，童写意早已成为众多新药研发英才的精神家园，成为中国新药思想的重要发源地之一。在这里，空乘服务是一道亮丽的风景线，抽象的报告会变得诗意盎然，枯燥的案例会化身成为舞台剧，经验的分享更是温情流露，感人至深。在这里，深邃的目光能看到世界新药动态的前沿。在这里，开阔的胸襟能在世界经济一体化的大潮中，明晰各国政府纷繁的药品注册法规。在这里，药物新靶点的发现、新工艺路线、临床试验，都成为了艺术，跃动在了科学家们的心间。正是这些有远见和使命感的新药研发英才，为我国的新药创制带来了勃勃生机。
借此机会呢，我对童写意新药英才俱乐部在15年来为中国生物医药创新发展做出的历史性贡献，表示衷心的祝贺。
国产 PD-1 抗体新药的相继上市，为癌症患者带来了生的希望。通过一致性评价的仿制药开始替代进口产品，惠及广大百姓。西达本案等自主知识产权新药的陆续获批，实现了从中国新药向全球新药的转变。与此同时，信达生物、君实生物、微星生物等一批优秀生物技术公司受到资本市场青睐，相继上市，让中国医药从制药大国走向制药强国，让我们的新药大国梦成为现实。这是童写意人的抱负，也是童写意人的实力，更是童写意人的使命。我们坚信，童写意人一定能绘出最壮美的新药画卷，让世界为之赞叹。